Thank you, Angelo. Good morning to everyone. Let me first start by thanking the organizers for putting the conference together and for selecting my abstract to present this work today. I'm indeed a postdoc in Justin Croker's lab at the EMBL at Heidelberg, but today I'll be talking to you mostly about work that I did in Edith Heard's lab back then at Institut Curie for my PhD, and some of which was recently published. So, as you've guessed already by the title of the talk, and my apologies, I will talk about TADs. So the general view on TADs and their relationship to gene regulation is that on the one hand, TADs help enhancers and promoters to come together and to meet in the physical space to regulate each other within the same TAD, while on the other hand, TAD boundaries prevent these enhancers from misregulating gene promoters in neighboring TADs, which is what happens in certain pathologies where TAD boundaries can be disrupted, and so these enhancers can ectopically regulate genes nearby. But what I want to discuss with you today is some of our work showing that this doesn't have to happen only in pathological contexts. So in non-pathological situations, we also have cases in which cis regulatory elements can communicate with genes in neighboring TADs, so across TAD boundaries. And just a disclaimer, with this arrow, I don't want to mean a physical contact or a contact as measured by chromosome conformation capture techniques, but just a functional interaction. So we've been exploring this at our favorite locus, the X-inactivation center on the X chromosome in mammals, where the exist gene resides. So exist produces a long non-coding RNA, which will be produced during development in placental mammals in embryos with 2 X chromosomes. And the exist RNA is able to coat the X chromosome from which it is produced in cis, and it will lead to its transcriptional silencing. So this process called X chromosome inactivation is thought to have evolved in placental mammals to equalize the dosage of X-linked gene products between XX and XY individuals. And so this process is critical. So if X inactivation doesn't happen during development of XX embryos, they die. So exist really needs to be very properly regulated for this process to take place as it should. So what do we know about regulation of exist? We know it's tightly coupled to the pluripotency network. So in the pluripotent state, in embryonic stem cells, including in vitro, exist is actually repressed. There's no expression even in XX cells, but as pluripotency factors go down during differentiation, exist expression can then happen, and as I said, from only one of the X chromosomes. Sometimes it happens on both, but then the cell needs to resolve this, otherwise it would die. But today, more importantly, I would like to talk about the cis regulation of the exist locus, and one of the critical cis regulators of exist is its antisense transcription unit. It's called TSIX, and TSIX is also regulated by the pluripotency network. It is actually expressed in the pluripotent state, both in XY and XX cells, and it is downregulated during differentiation, helping also to create a window of opportunity for exist to be expressed. We know that if we abolish TSIX transcription on one of the X chromosomes, during differentiations, these cells will undergo completely non-random X inactivation. So normally, exist can be upregulated from either one or the other X chromosome, but in TSIX heterozygous knockouts, exist will always be upregulated from the TSIX mutant chromosome, and this will be the inactive X chromosome. So TSIX negatively regulates exist in cis, preventing its upregulation, and there are other studies showing that it does so by its transcription across the exist locus, and in particular the exist promoter. So we know that other loci in the neighborhood of exist are also important for its regulation. So genetic dissection of single loci in the neighborhood have identified cis repressors and cis activators. And what you can appreciate is that they're actually spatially segregated. So you're probably already guessing how the TAD structure looks at this locus, we have two topologically associating domains, one in which the X promoter lies with its cis activators, while the TSIX promoter and other cis repressors lie in the neighboring TAD. Most genes within the TSIX TAD are also expressed in the pluripotent state, and they become downregulated during differentiation, while genes in the exist TAD mostly become upregulated with differentiation. So this TAD becomes more active during differentiation. So these two domains are not just structurally distinct, but they also seem to be functionally distinct. And so to explore this further, we decided to invert the exist TSIX locus, so we would place the exist promoter in the TSIX TAD and vice versa. So what happens, so if you look here at the capture C profiles, when we capture either the TSIX promoter or the exist promoter, you can appreciate that within this region, most of the interactions of the TSIX promoter fall within its own TAD, as we would expect, and the same is true for the exist promoter. When we invert the locus, now we switch the interaction profile. And so now the TSIX promoter is indeed, placed within the exist TAD, and most of its interactions happen within the exist TAD. So we've managed indeed to place them in each other's TAD, and from 5C analysis, you can appreciate that even though we switch their location, the overall TAD structure remains the same, pretty much the same. So still two TADs, and in general, the interactions are the same. So what happens in terms of expression? Does it matter? Well, yes and no. So when we looked at TSIX RNA levels, it seems to be pretty much insensitive to where, about its new location within the exist TAD. Whereas for exist, we actually saw massive upregulation even in XY cells, where normally, as you see here from the control, exist is not expressed or expressed at very low levels. And if we detect exist by RNA fish, we can also detect it. We can see either the pinpoint of the transcript, or in some proportion of cells, we even detect this exist RNA accumulation that is more typical of XX cells during differentiation. We generated this inversion also in XX cells, either in heterozygous or homozygous manner. And here as well, we saw misregulation of exist and TSIX. So precocious expression of exist, a higher number of cells where exist is expressed from both X chromosomes, which of course would lead to cell death if it's not resolved. And we also see that during late differentiation, TSIX expression, which normally goes down, is ectopically sustained. So clearly the topological partitioning of the X-inactivation center, so having each of these promoters in its own TAD environment, is critical to ensure the proper developmental timing of X-inactivation and the expression of these genes. And so this goes very much by the current views of how TADs and gene regulation operate together. The surprise came when we started dissecting the TSIX TAD, so on the left side, and we decided to delete most of the TAD except TSIX itself, its promoter, and a very proximal enhancer. But otherwise, we deleted all the other loci, so a region of 245 kb. So what did we see in this case? TSIX expression was affected, especially at later differentiation time points when it's sort of being downregulated. We could see that in the mutants, its expression was even lower. So there's some loss of expression of TSIX when its TAD is deleted. And for exist, we saw actually that in the mutants, and again, this is in XY cells where exist is normally not expressed, we could see high levels of exist. And again, by exist RNA fish, we could even detect some exist RNA clouds in a small proportion of cells. We generated this deletion in mice. And what you can appreciate from these results is that also in mouse, in female embryos, post-implantation, when X-inactivation is happening, we can detect a lot more exist RNA from the chromosome that harbors the deletion compared to the same black 6 chromosome in the wild-type embryos. So, okay, so what I've told you so far indeed goes along also with the current TAD views. So, you know, the TSIX TAD is important for TSIX expression. So when we delete the TSIX TAD, TSIX levels go down. And because TSIX regulates exist in Cs, TSIX goes down, so exist goes up. So this would be one scenario where, you know, whatever is happening within the TSIX TAD can have an impact on exist, but via TSIX, because there is this antisense transcription unit, and that would be the way to communicate from one TAD to the other. But now let's, for a second, imagine that the TSIX TAD could have elements that repress exist independently of TSIX. And so in that case, if we delete the TSIX TAD, we would see exist levels going up. And because the exist RNA can silence genes in Cs, it would be able to silence TSIX, and so we would see the TSIX levels going down. So actually, so far, we have no way to distinguish these two scenarios. Both are possible. And the second one would imply that there would be some communication between the TSIX TAD and the exist promoter in the neighboring TADs, so across a TAD boundary, which sort of goes against the current views. So to disentangle this, we made use of a system in which the exist and TSIX regulation is uncoupled from each other. So what these authors did was to knock in a GFP and a cherry reporter downstream of both exist and TSIX promoter on the same chromosome. And so the exist promoter cannot now generate any exist RNA, and the TSIX transcription is also abolished, so it doesn't repress exist in Cs. So they are uncoupled from each other, but at the same time, we can still follow their activity, or the promoter activity, by following GFP and cherry levels. So what we did was, in this cell line, to delete the TSIX TAD and ask, okay, how does it impact cherry and GFP levels now that exist and TSIX are uncoupled from each other? And so for cherry levels, which are a proxy for the TSIX promoter activity, we could see that they are down. So indeed, the TSIX TAD is important for TSIX expression directly, or at least independently of any effect on exist. But we saw the same thing for exist, or meaning in this mutant, when we delete the TSIX TAD in this cell line, we also see the GFP levels going up. And now, I remind you, this system, the exist is now completely uncoupled from TSIX. So this means that the TSIX TAD contains regulatory elements that negatively regulate exist promoter in a TSIX independent manner. So they can communicate with exist somehow across the TAD boundary. So we've tried to narrow down which elements within the TSIX TAD could be doing this job. And so we've dissected loci that previously had not been implicated in exist regulation. And we explored, 3 different CTCF clusters, 3 enhancer regions, the promoter of a coding gene and the promoter of a non-coding gene. And today I'll just have time to tell you about one of them. And this is the links-P element, as we called it. So actually, it's a 2kb element that overlaps with the promoter of a non-coding gene called links. So links RNA and the links locus is also active in pluripotent stem cells, pretty much like TSIX. So we were convinced that it would probably be an enhancer of TSIX. But when we deleted links, and we deleted it in different cell lines, but here I'll show you the data for this cell line where we have TSIX and exist uncoupled. When we delete links P in this cell line, actually we don't see any signs of it acting as an enhancer of TSIX. So the levels of cherry are pretty much similar. Actually even a bit upregulated. So what about exist? What we could see was that in the absence of the links promoter, these cells would more often express GFP during differentiation. So again, suggesting that links P can regulate exist in a TSIX independent manner. We deleted links P in mice as well, and in heterozygous female embryos, again, we could see exist RNA, so higher levels of exist from the chromosome where the links P is deleted. So, okay. We think then to have, how is it links P regulating exist then? And I don't have time to show you all the data, and we're not sure exactly how links P is working, but I can tell you what we excluded from a possible mechanism. So we did experiments with promoter inversion and poly-A knock-ins to show that these effects on exist are independent of links transcription and links RNA. So that's not how it's regulating exist. These effects are also independent of changes in topology, and we don't detect any privileged contacts between the links P element and the exist promoter. And when we did, gene expression analysis across these locus, but also by RNA-seq, there were no differentially expressed genes except links, because it's a knockout of its promoter, exist, which I showed you before, and CDX4, which is a neighboring gene of links, but we did knockouts of CDX4, and that doesn't have an impact on exist. So it doesn't seem to explain either how links is regulating exist. So we still don't know how exactly it's doing it, but all the genetic evidence shows that links P is a cis repressor of exist. We thought, I will just show you one more piece of data. So we thought of, okay, what if we would now place the links P element, which is acting as a cis repressor of exist, what if we place it inside the exist TAD? Would it still act as a cis repressor? And here we had another surprise. So we knocked in links P within the exist TAD at two different locations. I'm showing you here one, which was at the same distance, genomic distance of the exist promoter as in its native location. And so we knocked in links P on just one of the X chromosomes. So then we can ask, okay, is it preferentially expressed from that one? Or in this case, so if it's acting as a cis repressor, we would expect exist levels to go down. And we saw exactly the opposite. So when we clone links P within the exist TAD, it somehow acts now as an enhancer of the exist promoter, and we see the levels going up. And we see this regardless of the orientation of the links P element. Okay, so, well, I've just showed you that indeed communication between cis-regulatory elements and gene promoters in neighboring TADs is possible, apparently independent of privileged contacts between them, and we still don't know exactly how they are doing it, but we have recently identified another element within the TSIX TAD that also has an impact on exist regulation, and I'd like to mention some other studies that have also shown that this can be possible to have cross-regulation in neighboring TADs. So, of course, now one of the questions would be, does this happen despite of the TADs, this sort of regulation? Is it TAD independent, or is it, does it still depend on TAD structures somehow? And I would just like to finish with this idea that, there's other developmental regulated loci that lie at TAD boundaries or very close to TAD boundaries. And so we wonder whether these can actually be a way to regulate these genes, and often genes that have very exquisite regulation during development. And by being close to a TAD boundary and potentially having access to two different TADs, this can help in their regulation. So I don't know how I'm with time, but I would like to acknowledge everyone involved in these projects. Also our funding, and yeah, thank you very much for your attention. I look forward to your questions.
So in the system you use with the GFP in the M-cherry, where the transcription of T6 and exist are uncoupled, are the TADs looking the same?
We don't know. but we don't know in this system. I haven't checked, but I have done 5C on the TSIX knockout. So when we don't have TSIX transcription, and it doesn't look very different from the control.
Because somehow, I mean, intuitively, I would like to believe that the TADs are generated by the transcriptional activity and the whole regulation. So wherever you put the system, you will see similar folding patterns. And I don't know. Do when you put the links P element in a normal construct, not the uncoupled one, what happens?
When we insert in the exist TAD? In terms of structure, we also don't know what's happening there. Whether
and in terms of transcription, you see what?
So in terms of transcription, we only see effects on exist. Actually, the other loci within the TAD are not affected. Yeah, I just wanted to mention on your question. I also agree that some TAD boundaries seem to be driven by transcription. Our feeling here, from different dissections we've done, it's mostly driven by the CTCF sites. So there's a big cluster of CTCF sites at this TAD boundary.
So thanks, very interesting. So what are the histone modifications in the two TADs, and do you see a difference? Because presumably the element is only going to do what its histone modifications tell it to do.
Right. So it is. So this links P element is marked by active marks, because it in the pluripotent state it's a promoter, right? So it is marked by K27 acetylation. There's a strong peak there. So we haven't checked when we knock it in the exist TAD whether it keeps these chromatin marks. I would expect so, given also that it has an enhancer effect on exist expression. We haven't really profiled the chromatin marks in our mutants. In control cell lines, what we know is that the TSX TAD mostly has active chromatin marks in the pluripotent state, which disappear as the cells differentiate, because transcription goes down as well. And on the exist TAD, though, there's a strong K27 trimethylation enrichment, which with differentiation will evolve even into some K9. There will even be some K9 appearing there. So yes, it could be linked to that. My feeling also with all these different mutants is that somehow the exist promoter is very sensitive to the activity status of the TSIX TAD. Of the neighboring TAD. So, in this case, we lose links transcription, and this seems, exist sort of goes up. We have other mutants where, genes go up in the TSIX TAD, and then you see exist going down. So it's somehow sensing, but I'm still not sure exactly how, but sensing the activity status of the TSIX TAD.
Are only these two TADs that interact? So there is another TAD that act on exist? I don't know. Yeah.
There's one study suggesting some very long range interactions between exist and another non-coding RNA on the X chromosome, but far away. But these are a bit controversial studies. It's not very clear.
Looks like it's important.
It seems to be more important after these initial stages of X-inactivation, so it seems to be more important to maintain exist expression once it starts during differentiation.
Thank you.